link between gastric cancer and H. pylori infection. The recognition of H. pylori is an important factor in gastric ulcers took many years to gain acceptance. Even in my short 25 years, it was not accepted in the beginning. So I'm glad to see this progress and these kind of studies coming out that kind of back up what we had been saying for quite some time. And one would hope that the significant role it seems to play in gastric cancer will be recognized much more quickly after studies and programs like this. So, Dr. Aron, I'd like you to take it away.
Thanks, Reed. In each one of these sessions, as I always say, we take a deep dive into recent research that brings to light objective scientific evidence, and that's what we're looking for whatever the topic is that we're dealing with. And today, let's look more closely, as Reed said, the mechanistic link between gastric cancer and H. pylori infection. It's a very detailed study, and I hope that we'll be able to give you sort of the Cole's Notes version of what they were looking at. We know that chronic disruption of mucosal barriers, together with ulcerations, etc., will predispose to greater risks of malignant changes going forward. So let's look at the actual evidence, because science matters.
Thank you, Dr. Gonshor. Right before we get into this new topic, in our last broadcast, we discussed the SIG-A regulation of pathogenic gut fungi and its dysregulation in Crohn's disease. Fascinating, kind of tied to today's discussion, which also involves a healthy mucosal barrier. And there were a number of questions, Dr. Gonshor, including this one. Why is it so important to test secretory IgA in saliva?
Well, because we're trying to keep this down to a few minutes, we could spend a whole day on this, but the usefulness of testing secretory or secretory IgA in saliva really lies in assessing the function of the immune system, both locally, whether it's in the oral cavity, in the gut, or in the entire immune system. Because remember, it's the first line of defense. So elevated SIG-A levels may indicate a very strong immune response to a particular antigen or a pathogen of one kind or another, while a decreased SIG-A level is indicative of reduced mucosal immunity. And that's not a good sign, folks, because when that happens, that means that that first line of defense has broken down. And you can expect that a lot of pathogens are then able to get to the intestinal cell, beyond that, into the submucosa, and now you're into a whole different ballgame of what the body needs to do. And it has to bring out the big guns, and that is not what you want. So in the causes of elevated SIG-A, that would include a whole number of things. A defective epithelial barrier, that one, in the case of the gut, it's a one-cell barrier, which allows bacterial and microbial penetration. It is the strongest stimulator of SIG-A production. There are also consistent elevations in those, by the way, who have celiac disease, colon cancer, IBS, which is irritable bowel syndrome, which is a whole other story. And especially in the case of IBS, those who have the diarrhea subtype, and we can spend one of our other broadcasts in that area. So please note, when you're doing an analysis of Candida in the gut, it is critical to know what amount is hyphal. If you remember what we did last time, it's not the commensals we're as concerned about as the hyphal form. That's what's the aggressive form. So we want to know how much of the Candida is pathogenic, how much of it is really able to penetrate the mucosal barriers. That's what you want to know. That test does not exist today. And that's why we stopped doing the Candida test that we were doing, because it wasn't meeting the actual needs. Here's the good news. We're working on it, and we hope to have a test available in the next 6 months, which is going to show you how much of the Candida is spherical, meaning commensal, how much is hyphal, what is the ratio of the two, and bingo, you're going to be able to have a very clear look as a screening test whether there's something to be worried about. So we're going to be putting that together for you very, very soon.
And we're going to hold you to that, Dr. Gonshor.
Yeah.
We love our testing.
Boy,
and we've been talking for decades about the yeast, the fungi, and especially candida, and whether it is invasive or not. Kind of this, so having that test to clarify what's commensal, what's invasive, or an overgrowth, a harmful overgrowth, will be really, really handy. So I want to thank everyone for submitting your questions. Really important to all of us as we learn together. Now get your pens and your paper ready, because Dr. Gonshor is going to jump in to today's study about mechanistic link between gastric cancer and H. pylori infection. Dr. Gonshor.
Well, thank you, Reed. So today's broadcast is dealing with mechanistic link between gastric cancer and H. pylori infection. And we're looking at this particular study, which literally just came out several months ago. Very, very large group of people. The Helicobacter pylori Cas-A oncoprotein disrupts WNT PCP. I'm not even continue. It's a very long title. What does it tell you? There is an oncoprotein, meaning a protein that can cause malignant changes. It's called Cas-A, and it may contribute to the development of gastric cancer. And how does it do it? By subverting a mechanism that normally restrains pyloric gland stem cell proliferation, and it promotes enteroendocrine differentiation. So we're going to get into that in much more detail. But one of the things you need to remember is the following. As we said, H. pylori infection for many decades was poo-pooed as a reason for gastric ulcers. It took an awful lot of work to finally get it accepted, and today it is accepted that H. pylori, which enters the mucus layer of the stomach, it attaches to the stomach lining, it causes increased acid production, it damages the stomach lining, and in fact can cause peptic ulcers. In fact, it's the main cause of peptic ulcers, as well as various types of gastritis, and as it turns out, stomach cancer. And fully 30 or 40% of people in the United States are going to get some form of H. pylori disturbance during their lifetime, 30 or 40%. So that's a very, very large percentage. But what about the issue at hand with gastric cancer? Well, there's clinical evidence now that infection with a particular type of H. pylori, it's the H. pylori Cas-A plus strains, dramatically increase the risk of developing gastric cancer. What's the mechanism of that? Well, turns out that this particular oncogenic Cas-A, remember the type of genetic group within H. pylori that can cause malignant changes, it may promote gastric cancer development. And how does it do it? It disrupts a particular pathway, which is the WNT/PCP pathway, that normally restrains pyloric gland stem cell proliferation, and it promotes enteroendocrine differentiation. And what does that mean? Well, H. pylori infections commonly are associated with abdominal pain, bloating, acidity, but infections with this particular strain, the Cas-A strain, is also the strongest risk factor for gastric cancer. Now, Cas-A, it's a gene that codes for an immunodominant antigen. So it takes over antigenically. So the H. pylori begins to become effectively a factor in gastric cancer creation. It's present only in H. pylori strains that are associated with severe gastroduodenal disease. In other words, not all of them, thank God, not all H. pylori is like that. But when you have severe gastroduodenal disease, it begins to show its ugly head. Cas-A, then, when delivered into gastric epithelial cells, it acts in effect as a pathogenic scaffold. and it functionally disturbs a whole bunch of the host proteins to do what? To promote gastric carcinogenesis. It disrupts this particular pathway that I told you about, this WNT pathway. It's a signal transduction pathway. It's made of proteins, and they pass signals into a cell through various cell receptors. And this particular CAG-A oncoprotein, it's a cytotoxic associated gene A. It's delivered by certain H. pylori strains into gastric epithelial cells. And what it does is it causes not only gastric disease, but also gastric cancer. And the way that it's doing it is that this CAG-A group dampens the proper formation of stem cell niches. And you can see the typical niche in a pyloric gland. These are stem cells that are producing the various types of endocrine cells, which are sitting in the pyloric glands. And it creates hyperplastic changes in the pyloric glands. Just many more are differentiating. A tremendous amount of these stem cells are producing these pyloric glands cells, and that's not what you want, because if you start to accumulate too many of them, then eventually some of them are going to go rogue and they're going to become pathogenic, leading to possible gastric cancer. So the conclusions, the combined results showed the following: molecular mechanisms that are involved in gastric carcinogenesis are induced by what? By H. pylori. How? Through this WNT PCP pathway, which plays a very important role in carcinogenesis. And they propose that this pathway is a potential target for clinical intervention against pylori CAG-A infections, because if you can target this particular pathway, you may be able to stop some of the worst consequences. So here's a quote: perturbations or a whole series of changes in this particular pathway induces hyperplastic changes in gastric pyloric glands, and that may contribute to the development of gastric cancer, in conjunction, of course, with other oncogenic CAG-A actions. It's only one of a number. We're beginning to see that this signaling pathway is quite important. So what are the key takeaways? The oncoprotein CAG-A, which is delivered by certain H. pylori strains into gastric epithelial cells, causes gastric diseases, including, of course, the unwanted gastric cancer. What is proposed? That it is this particular WNT PCP signal pathway which may be a potential target for prevention of gastric cancer, which is developed by these H. pylori CAG-A strains. And with that, we thank you. This is a very truncated explanation.
Well, thanks for that, Dr. Gonshor. You did simplify what I found to be a pretty difficult study. Some of them I can get through more easily than others. This one was a little bit on the hard side, but what I appreciated was that it helped me understand the virulence factors, because the test that we use for H. pylori does not speciate. There's different varieties of H. pylori. And of course, it's this CAG-A, and there's the VAC-A, and there's the others. There's the BABA or BAB-A, and others. And this study, as difficult as it was for me to get through, really helped explain that. And of course, you made it even easier. So thank you for that. And we're going to have some questions come in, I'm sure. And thank everyone for submitting questions. We've done the study, and we've got our key takeaways. And if you enjoyed this episode, please like, subscribe, share, and all those sorts of things. You can submit your questions, comments, and suggested topics to fdntraining.com/science matters. And of course, grab a copy of the studies at fdntraining.com/studies. We've got quite an archive going. And join us again in 2 weeks on November 1st when we talk about the the C gas sting, a signaling pathway which mediates immune sensing of DNA, drives age-related inflammation and neurodegeneration. And why do we do this? Because
Science matters.